The passage we're going to discuss now is a passage based on biology. It's based on an experiment done on octopuses. How do octopuses behave when they're immersed in a solution of the drug ecstasy? The basic passage says that there was not the octopuses became more sociable after when they were dipped or when they were given octopuses. All right. So we will look at the passage, we'll summarize it quickly and discuss the questions. When Girl Darlene first gave ecstasy to octopuses, she didn't know what to expect. She's a neuroscientist at the Johns Hopkins School of Medicine who studies how the cells and chemicals in animal brains influence animal social life. Ecstasy, also known as MDMA, interests her because it's known to make people feel more sociable, more interested in others, and less defensive. She and her colleagues, Eric Edsinger, put 5 Californian, so they conducted an experiment. They wanted to say, how does ecstasy affect octopuses? Why did they want to do this? Well, it affects human beings. It makes them more sociable, more friendly, less defensive. So therefore, she wanted to conduct an experiment on octopuses. She and her colleagues put 5 Californian 2-spot octopuses, that's a particular species of octopus, individually into the middle of 3 connected chambers. So 2 octopuses, she conducted this experiment on 5 octopuses individually. And she put these octopuses individually into the middle of 3 connected chambers. So she took 5 of them and put 1 of them individually, one by one, in different spots. I mean, the experiment was conducted on 5 of them. But at a time, she used to put 1 individually into the middle of 3 connected chambers. And she gave them full rein to move around in the 3 chambers. All right, okay. So there are 3 chambers. She has introduced the octopus into the middle chamber. One of the adjacent chamber housed a second octopus, all right, confined inside an overturned plastic bucket. The other contained an unfamiliar object such as a plastic flower or a Chewbacca figurine. Darlene and Edsinger measured how long the main animal spent in the company of its peer and how long with the random toy. So this is the setup of the experiment. Chose 5 octopuses, left them one by one individually, conducted the experiments by leaving each individually into 3 chambers, 3 interconnected chambers. The middle one was where this octopus was let go. The octopus was free to move around in all the 3 chambers. One of the chambers had another octopus. That second octopus was confined in a overturned plastic bucket. This octopus could go there, but that couldn't come. And the other chamber had a random toy, some plastic flower or some unfamiliar object. So this is the experiment. What were they looking for? What were they measuring? How long the main animal spent in the company of its peer, which is the main animal, the experimental octopus, and how long with the random toy. All right. This is exactly the kind of setup that neuroscientists use to test social behavior in mice. But Darlene had no idea whether it would work with octopuses. It might be that they are so smart that this kind of task that we would use for a mouse would be boring to them. Maybe they'd take 1 lap around the chambers and stop. Fortunately, that's not the case. So they do a similar experiment with mice. And Darlene said, would an octopus also respond the way mice do, or because an octopus is way more intelligent, they'd get bored by these games and not move at all. Fortunately, the octopus did respond. The free-moving individuals thoroughly explored the chambers, and from their movements, Darlene realized that individuals of any sex gravitate towards females but avoid males. All right, this is one. What did they observe? They moved around freely. What is the second thing they observed? Individuals of any sex, whether the main octopus you are letting roam around the three chambers, whether that's a male or a female, it didn't matter. It gravitated only towards the female and it avoided the male. Clear? So these are the two. These are what was observed. Next, she dosed the animals with ecstasy. Okay, this was done. Next, she dosed the animals with ecstasy. This drug, she put the animals. Again, there's no precedent for this. And how do you know? How do you give ecstasy to an octopus? There's no precedent for this, but the researchers often anesthetize octopus by dunking them in ethanol, a human procedure with no lasting side effects. So Dolan and Etzinger submerged their octopuses in an MDMA solution. So how do you introduce this drug into an octopus? Well, they said that when they wanted to anesthetize octopuses, they just immerse them in ethanol solution. So the same way they immerse the octopuses in MDMA, which is the other name of ecstasy solution, allowing them to absorb the drugs through their gills. At first, they used too high a dose, and the animals freaked out as did all these color changes. But once the team found a more suitable dose, the animals behaved more calmly and more sociably. So there were a few hits and misses, trial and errors in this experiment, but they finally found the right dose which made the animals behave a little calmly, and then they could observe it. With ecstasy in their system, the 5 octopuses spent far more time in the company of the same trapped male they once shunned. This is the difference. Normally, when they let the octopuses went more towards females. Whatever sex the octopus that was released, it avoided the male octopus. But after dunking in ecstasy or MDMA, the 5 octopuses spent more time in the company of the same trapped male they once shunned. Even without a stopwatch, the change was obvious. Before the drug, they explored the chamber with octopuses very tentatively, not very sure of themselves, unsurely. They mashed themselves against one wall, very slowly extended one arm, touched the other animal, and went back to the other side. So very tentative movements, very unsure, uncertain movements, Dolan says. But when they had MDMA, they had this very relaxed posture. They floated around, they wrapped their arms around the chamber, they interacted with the other octopus in a much more fluid and generous way. They even exposed their underside where their mouth is, which is not something octopuses usually do. They do not reveal their underside where their mouth is, but they even did that. So this is the way the octopuses reacted when they had ecstasy. But, so here we have an experiment, we have the observations of the experiment. Now we come to but. But always shows the other side, the other point of view. Jennifer Mather from the University of Lethbridge isn't convinced that ecstasy is making them sociable. Instead, the drug might just miss their ability to detect the chemical cues of potential mate. There's no proof that it is anything more than attraction. This may not be the effect of the drug at all. This is one of the scholars from the University of Lethbridge. Harriet DeWitt from the University of Chicago, another lady from another university. The scholars from one university conducted the tests. They must have published their reports in a scientific journal, and the other two have read those observations, and they are saying, no, listen, this change may not, so one of them says, may not be the effect of the drug at all, it must be just natural attraction. Second one says, from the University of Chicago, Harriet DeWitt, who has studied the effect of ecstasy's effects on animals, had other concerns. What are her concerns? It's an innovating and exciting study, she agrees. But it's unfortunate that the duo always tested the octopuses first, after a dunk in normal salt water and then after an ecstasy bath. She said the procedure, she had an objection to the procedure they followed. They first dunked the octopus in salt solution and then observed it, and then they gave it an ethanol bath. So she had a problem with that. In pharmacy studies, scientists normally mix up the order in which the animals received the drug and the saline control. The saltwater dunking is the saline control. So normally the procedure followed by this octopus study was first salt water, then the MDMA. Normally scientists mix up the order. So this is the objection that the scholar from University of Chicago had. She said, why did they first dunk it always in salt and then in MDMA? Without that counterbalancing, so what is the root, what is the cause of her objection? We'll find the explanation now. Without that counterbalancing, it's hard to say why the octopuses were behaving differently the second time round. So without that order, we don't know why the octopuses were behaving differently. So she has come up with her own explanations. Was it because of the ecstasy that they behaved differently? Or was it because they had become familiar with the arena, the plastic toy and other octopuses? After all, it's a question of familiarity. Once you've given them time, then next time that fear, the shyness and all will go. So did they react this way, respond this way because the effects of ecstasy, or was it just familiarity of their situation? That's what she's asking. The objection raised by Harriet DeWitt from the University of Chicago is based on which of the following? Harriet DeWitt, University of Chicago, was given in the last paragraph of the passage. What did she say? She said it is that exciting experiment, but the experiment always followed the same order. First salt water and then MDMA. Because of this, there was no counterbalancing happening. So we don't know if the octopuses behave differently because of the effect of ecstasy. It's not clear. Or because they are familiar. Suddenly, once they've spent time dunked in salt water, then again the ethanol. So now they've got used to the conditions of the experiment. So we don't know whether their change in behavior was a result of ecstasy or their familiarity with the surroundings. So we will look at an option which is closest to that answer. The drug used in the experiment has been diluted. No, it didn't say anything about dilution or concentration of the drug. The octopus that was involved could have become habituated to the drug. She said familiarity with the surroundings. She didn't say habituated to the drug. The octopus could have become familiar with the surroundings of the 3 chambers and everything there. Yes, that is correct. The drug had very little to do. She didn't say it. She said it's not clear. Is it the drug or is it familiarity? Therefore, my correct answer is C.